Welcome everybody to this presentation on cosmetic testing and FDA regulatory compliance. So in today's agenda, we're going to be covering various topics concerning cosmetic testing and how dealing with the quality and safety of cosmetics, so we can see how FDA is, FDA's approach towards regulation of cosmetics. So we're going to be talking about microbiological testing for cosmetics suggested by FDA. We'll be looking at animal testing and cosmetics, various contaminants in cosmetics. We'll be looking at color additives that are used in cosmetics, as well as batch certification of certain color additives, different ingredients that are prohibited or restricted. We'll also be discussing expiration dating or the shelf life of cosmetics, as well as nanomaterials and good manufacturing practices in cosmetic manufacturing. So the prime goal of FDA is to ensure that cosmetics when on the shelf for the consumer are safe for the consumer to use, in accordance with the label directions or as may be expected, from the label and packaging. So cosmetics should not be misbranded, they must not be adulterated, and they must be prepared, packed, and stored in a way that ensures that there's no contamination or health risks. So to prevent contamination, microbial contamination can occur in various ways. So you can have raw materials, water, or other ingredients that could get contaminated. Manufacturing conditions could be a root cause of microbial contamination. You could also have various ingredients within the cosmetic that promotes microbial growth, especially when not used with a good preservative. Packaging and storage may not be adequate. There may be poor shipping conditions. And then lastly, consumers can also cause contamination of the end product, for instance, with samples that are on a shelf. So to deal with the various areas of microbial contamination, FDA has their own bacteriological analytical manual, and this manual, Chapter 23 of the manual, discusses microbiological methods for cosmetics, and this was recently updated in July 2017, in specific areas. But overall, the manual discusses various methods. So methods to isolate microorganisms, like direct colony counts, enrichment culturing, and those isolation procedures could be used to treat water-insoluble products and render them miscible. There is also a lot of discussion around media preparation, including dilution and plating of media, and media preparation to partially inactive preservative systems are commonly found in those cosmetic products, and so media is used to there are various dilution and plating media techniques around that. There are also identification of isolated microorganisms by routine microbiological methods or by commercial identification kits that I discussed, as well as a scheme proposed for the enumeration, isolation, and identification of various cosmetic microorganisms. So media is prepared for the enumeration and identification of gram-positive bacteria and fungi, and the different kinds of media preparation are discussed in this manual. There is also discussion around media preparation for the identification of Enterobacteriaceae, as well as for the identification of gram-negative non-fermentative bacilli. The manual also discusses handling of samples for microbiological analysis, including preliminary sample preparation, be it liquids, powders, solids, oil-based and clean products, or aerosols of powders, soaps, liquids, and other materials like including anhydrous materials. There are different microbiological evaluation procedures that are detailed in the manual, like the aerobic plate count, the fungi, yeast, and mold plate count, as well as anaerobic plate count, which is used for talcs and powders. Different microbial identification methods, like the cultivation method and microscopic examination, are used to identify gram-positive rods. Then the catalase test and the coagulase test are used to identify gram-positive cocci, and there are multiple agar that are discussed, that are used to identify gram-negative rods. Pseudomonas aeruginosa is another microorganism that requires identification, and there are different tests like the TSI, agar salons, and the oxidase tests that are used and suggested by the manual for that organism's identification. And different biochemical tests are also described, including tests involving YE agar, co-surge citrate broth, malonate broth, and the nitrate mobility agar. Additional media that are used in biochemical tests include the arginine T-carboxylase broth, the gelatin liquefaction, Pseudomonas agar F, Pseudomonas agar P, as well as tests for flagellaceae. And then there's also preservative efficacy that is, tests for cosmetic preservative efficacy that is also detailed in the manual. Animal testing. So with respect to animal testing, there's no specific requirement to test animals in, to ensure cosmetic safety. However, animal testing can be used by manufacturers to establish cosmetic safety, and manufacturers need to be a bit careful when they're doing animal testing. They need to ensure that it's appropriate and effective to substantiate cosmetic safety, and animal testing needs to be done in the most humane manner, and so that means using the smallest number of animals and and deriving the maximum amount of scientific information from the smallest number of animals to test, to test cosmetics. So there are various laws and regulations and policies that do govern animal testing, and they include the Animal Welfare Act and the Public Health Service Policy of Humane Care and Use of Laboratory Animals. So FDA, while animal testing can be done, FDA recommends alternatives, and these alternative methods to whole animal testing have been suggested by the NTP-ICC-BAM, which stands for the National Toxicology Program Interagency Coordinating Committee on the Validation of Alternative Test Methods. So this committee really coordinates the development, validation, acceptance, and harmonization of alternative toxicological test methods. throughout the U.S. So, therefore, these alternative validated methods are suggested instead of animal testing. And again, FDA suggests limiting any animal testing if need be to the minimum number of animals, and that can be used to derive the maximum amount of scientific data. What about claims like cruelty-free or not tested on animals? So there is no real legal definition for these claims or these terms, and manufacturers have relied on raw material suppliers or contract labs to do the animal testing to substantiate product or ingredient safety. So in those cases, they might be using these claims. Additionally, they might be using a combination of non-animal testing or raw material safety testing coupled with scientific literature or even controlled human use testing. So in these situations, these manufacturers may be using these claims like cruelty-free, not tested. And this, many years ago, raw materials were tested on animals when they were first introduced, and so now, it's possible that these cruelty-free claims are based on the fact that the materials or products are not currently tested on animals. That could also be another case scenario when these claims are used in labeling. So moving on to potential contaminants identified by the FDA, so you need to be wary of these in the cosmetic industry. So one of the potential contaminants is 1,4-dioxane. Now, 1,4-dioxane is a contaminant that is present in extremely small amounts in some cosmetics, and it is a byproduct of the manufacturing process of certain cosmetic ingredients. In the 1970s, the National Cancer Institute found that there was an association between 1,4-dioxane and animal cancer. This ingredient's been shown to penetrate animal and human skin when it's applied in certain preparations like in lotions, but it does evaporate easily. So there's no specific limit set by FDA for 1,4-dioxane, but FDA is monitoring this ingredient as a potential contaminant, and so is recommended vacuum stripping process at the end of the polymerization process to ensure safety. BSE, or as more commonly known, mad cow disease. BSE stands for bovine spongiform encephalopathy, and this is a potential contaminant, identified by FDA that could occur in cosmetics. BSE is, a progressive neurological disorder of cattle, and it's thought to be caused by a modified cell protein called a prion. And when cattle are fed by fed meat and bone meal, they're prone to getting BSE. So the BSE agent has been found in cattle brain tissue, in the spinal cord, the eye retina, small intestines, the tonsils, bone marrow, and the dorsal root ganglia along the vertebral column. So BSE is not the only progressive degenerative neurological disease. There is actually a group of them called transmissible spongiform encephalopathies, or TSCs, and there are 6 of these diseases, or TSC diseases, that affect humans, and they're extremely rare, but they can be fatal. So these 6 diseases include Kuru, classical Creutzfeldt-Jakob disease, CJD, Variant Creutzfeldt-Jakob disease, VCJD, Gustafson-Straussler-Scheinker syndrome, fatal familial insomnia, and sporadic fatal insomnia. So food can be contaminated by BSE, and there's a lot of evidence, epidemiologic and lab evidence that suggests a new variant, Creutzfeldt-Jakob disease and bovine spongiform encephalopathy are caused by the same infectious agent. So what was the response by the federal government in the U.S.? So the U.S. Department of Agriculture, which is responsible for the livestock health, has prohibited, the import of live ruminants from countries where BSE, is in their native cattle. And on the health, the cosmetic product side, FDA has prohibited the use of most mammalian protein in the manufacture of animal feeds that are given to ruminants. This is to stop, the spread of this potential contaminant to other products like foods and cosmetics. So just as a bit of an aside, a little bit more on what the U.S. Department of Agriculture is doing. They have a final rule where they remove downer animals and specified risk materials and tissues from the human food chain. They require additional process controls for establishments using advanced meat recovery. They hold meat from cattle that are targeted for BSE surveillance testing until the test is confirmed that there's negative, no BSE present, and they prohibit air injection, stunning of cattle. So FDA, for their side, has prohibited various cattle materials. These include specified risk materials, and those specified risk materials, or SRMs, include the brain, the skull, the eyes, the trigeminal ganglia, the spinal cord, the vertebral column, excluding the vertebrae of the tail, the transverse processes of the thoracic and lumbar vertebrae, and the wings of the sacrum. Also included as a specified risk material is the dorsal root ganglia of cattle that are 30 months of age and up, and the tonsils and distal ileum of the small intestine from cattle. Now, the small intestine is prohibited. There is an exception to that, and that is when the distal ileum has been properly removed. Other prohibited material include material from non-ambulatory disabled cattle, as well as material from cattle that have not been inspected or that have not passed inspection or mechanically separated. Now, there are certain products or ingredients that are not prohibited, and but there are so there are limits. For instance, milk and milk products, high derived products, and tallow that contains no more than 0.15% of insoluble impurities, as well as tallow derivatives. These are not prohibited cattle materials. Gelatin is also not prohibited as long as it's manufactured using customary industrial processes as specified in FDA's final rule. So it is on the onus of manufacturers to ensure that all material is from the cattle is not using prohibited cattle materials, and manufacturers need to ensure that they have the records on file that sufficiently demonstrate that their cosmetics are manufactured and that they are free from prohibited cattle materials. And this is to ensure that there is no potential for BSE. Lead is another potential contaminant. There is a risk of lead poisoning, and this is serious in the case of children because they are susceptible to absorbing lead from the environment. So with high levels of lead exposure, there can be serious problems like anemia, kidney issues, neurological damage like seizures, coma, and death. With low levels and chronic exposure, there could be learning and behavioral problems. So lead as a color additive in cosmetics must therefore conform to the color additive regulation of FDA. This is typically not more than 20 parts per million. Some other ingredients like coal, kajal, alcohol, or surma. So in these lead, sometimes accounts for more than half the weight of sample of coal, and this is usually in the form of lead sulfide. Therefore, there is an import alert that is in effect for cosmetics containing coal, and basically there is detention of these products without, physical examination of eye area cosmetics that contain, these coal, kajal, surma. Lipsticks, hundreds of lipsticks have been analyzed by the FDA for potential lead contamination, and FDA found that there is, a very low health risk potential, and so there isn't too much concern concerning lipsticks. However, FDA has suggested a specific method for determining the total lead in lipstick, and this has been suggested to industry to use. This method is publicly available in our journal, online at the link below, and the method is development and validation of a microwave-assisted digestion, inductively coupled plasma mass spectrometric method. So you can visit this journal and refer to the method to use FDA's suggested method for analyzing lipstick in lead. Now, lead acetate is used as a color additive in some progressive hair dyes, and when it is used in such a manner, it is subject to FDA's color additive regulation, and this includes the specifications. Usually all color additive regulations, they include a specification section, uses and restrictions for that color additive, as well as any cautionary statements that would apply as determined by the FDA. So in this case, with respect to progressive hair dyes, the lead acetate limit is less than or equal to 0.6% of lead, and there is a cautionary statement that is required in the labeling, and that should read: caution, contains lead acetate for external use only, keep this product out of children's reach, don't use on cut or braided scalp, and if there is irritation, discontinue, don't use it on the mustache, the eyelash, eyebrows, hair, or on hair parts of the body. Other than the scalp, don't get it into the eyes, follow the instructions, wash hands thoroughly after use. So just a cautionary statement that is required in the labeling. Now, all color additives require FDA approval, and they must conform to FDA's regulations, FDA's color additive regulations, which specify the specifications, the identity of the cosmetic, the uses and restrictions, as well as other labeling requirements like cautionary statements. So their color additives can be grouped into different categories, and these include lakes, straight colors, and then colors that are subject to certification. So this is usually called batch certification, and then those that are exempt from batch certification. So let's discuss what we mean by color batch certification. So color batch certification is required for a specific group of color additives, and those are the color additives that are subject to batch certification. Now, FDA does analysis on a batch of color additive, and then provides a unique lot certification number. And this FDA lot certification number indicates that the batch in question has successfully passed FDA's analysis of its composition and purity in FDA's own labs. So that's what FDA batch certification of color additives is really all about. And it is required before the color additive can be used in the cosmetic. And there are some FDA fees, and that is really based on batch weight. So there are different kinds of requests that can be made of the FDA, different filings, and they include a request for certification of a batch of straight color additive. You can see it's sometimes grouped by the category of the color additive. So there's a request for certification of a batch of color additive lake, and there's also a request for certification of a repack of a batch of certified color additive, or a request for certification of a batch of color additive mixture. So different kinds of packaging. So, for ingredient safety now, cosmetic ingredients do require to comply with FDA's compliance requirements with respect to certain ingredients. And they could be precautionary labeling statements, like a cautionary statement, or it could be specific concentration limits, compliance with analytical specifications, test specifications, limits to use of those ingredients, or restrictions. And there could be also ingredients that are completely prohibited on FDA's prohibited ingredients list, in which case you cannot use those ingredients. So let's discuss some select cosmetic ingredients and discuss FDA compliance requirements around those ingredients. So one is alpha hydroxy acids. Now, with respect to AHAs or alpha hydroxy acids, there is a sunburn alert that is required by FDA, and that should state that the alpha hydroxy acid basically can increase the skin sensitivity to the sun, and so it should advise consumers to wear sunscreen or some protective wear, and limit sun exposure when using the product, and for a week thereafter. Now, in terms of concentration limits, AHA limit is 10% or less in a cosmetic, and the final product should have a pH of 3.5 or more, and it should also be formulated in such a way that it protects the skin from increased sensitivity, or packaging directions should direct consumers to protect themselves from the sun. Now, talc, because of its close proximity to asbestos in the earth, there has been discussion of whether talc could be a carcinogen. And however, FDA surveys have found that there's no asbestos detected in cosmetic grade raw material, talc. So other ingredients are also monitored by FDA because of some association at some point in time or the other, and so FDA continues to monitor these ingredients. So they include parabens because of their association with breast cancer, fragrances, and allergies have been found to be associated together. There are also beta-hydroxy acids, BHAs, and these include your salicylic acid, beta-hydroxybutanoic acid, tropic acid, ethylcanic acid, and these have been associated with sunburn. Diethanolamine, DEA, that are used as emulsifiers and foaming agents, have been found to cause cancer in lab animals in a 98 study. And phthalates have unknown adverse health effects. They may have unknown adverse health effects, so they're monitored by the FDA. Now, some other ingredients are prohibited, and they include bipinyl because of photo contact sensitization. Chlorofluorocarbon propellants are prohibited in cosmetic aerosols that are used for domestic consumption. Chloroform has been associated with cancer in animals, and so it's prohibited. The exception to that is when there are very residual amounts from its use as a processing solvent during manufacture or as a byproduct from the synthesis of an ingredient. Halogenated salicyl anilides like ditribromosalin and tetrachlorosalicyl anilide are prohibited because they've been known to cause serious skin disorders. Hexachlorophene or hexachlorophene is a restricted ingredient, and that's because of its ability to penetrate the skin, and it's toxic, and so it's restricted to not more than 0.1% concentration, and it should, any labeling should say that it's not to use in mucous membranes like the lips. And then some other restricted or prohibited ingredients are mercury compounds because of their easy skin absorption and accumulation in the body, and they're known to, cause skin irritation or allergic reactions or other neurotoxic issues. They are restricted to not more than 65 parts per million for eye cosmetics, less than 1 part per million for other cosmetics, and they're subject to manufacturing conditions that comply with FDA's cosmetic good manufacturing practices, which we will be discussing. And for another ingredient, methylene chloride, this is prohibited because it's shown to cause cancer in animals. Prohibited cattle materials, we've discussed this. They are included in the list of FDA's prohibited ingredients. Sunscreens and cosmetics. Sunscreens are not cosmetics. They are considered drugs and are subject to FDA's more stringent drug regulations. And vinyl chloride is prohibited in aerosol products because it's shown to cause cancer and other health issues. Zirconium-containing complexes are prohibited in aerosol cosmetic products because of their toxicity to animal lungs, and there's been formation of granulomas in human skin by exposure to these ingredients. So shelf life or expiration dating. Now, with respect to a cosmetic, there is no set regulations on this. However, manufacturers have to determine the shelf life to ensure that the cosmetic, is safe for the entire shelf life. So it's the manufacturer's responsibility to ensure that their cosmetics will be safe for the entire period, and so they need to set a shelf life. They should do so. The main objective here is to ensure that the cosmetic is not misbranded and is not considered adulterated by the FDA. So there's no potential for contamination or other health risks. So various factors can affect the shelf life. So they can be, during manufacture or when they're out on the shelf available to consumers. So, for instance, plant-derived substances in a cosmetic can be, conducive to microbial growth, and this can be a problem. This can limit shelf life in cosmetics that are considered, all natural. Then there is also, lack of preservatives or non-traditional preservatives that could increase the risk of contamination. Reusable samples is, a very good source of, potential contamination. Also deterioration because of climate conditions like a high temperature could also limit the shelf life of the cosmetic. So what should a manufacturer consider? While there aren't any regulations, there are some guidelines. So there are some articles. So for instance, there was one in 1980 which suggested 18 to 24 months, but that shelf life was really to maximize cost efficiency in warehousing, distribution, and marketing. Another, article by John J. Cabara in 1984 suggested regularly evaluating samples in product stability, so doing stability testing for 3 years or more, depending on the product. And the EU, the EU's cosmetic derivative has suggested expiration dating for products whose minimum durability is less than 30 months. Nanomaterials. So there have been, some nanomaterials and cosmetics is an evolving field, and so far there is some discussion about nanomaterials and its use of cosmetics. So let's discuss nanomaterials first of all. Nanomaterials, they can have physical, chemical, or biological properties that differ from those of large-scale material with the same chemical composition. They can vary widely in composition or morphology or other characteristics, and so they cannot be considered a really uniform group of substances. So there can be differences that include altered magnetic properties, altered electrical or optical activity, increased structural integrity, or altered chemical or biological activity. So what is a nanomaterial then? So a nanomaterial is considered to be defined as a material or an end product is engineered to have at least one external dimension or an internal surface structure in the nanoscale range. So we're talking about 1 nanometer to 100 nanometers. A nanomaterial can also be a material or end product that is engineered to exhibit properties or phenomena that include physical or chemical properties or biological effects that are attributable to its dimensions, even if these dimensions fall outside the nanoscale range. And this would be up to, though, 1 micrometer, so up to 1,000 nanometers. And nanomaterials can be grouped. So then they can be grouped into soluble and biodegradable nanoparticles that disintegrate into molecular components like some liposomes and nanoemulsions, or they can be insoluble and sufficiently stable or biopersistent particles like titanium dioxide, fullerenes, and quantum dots. So how do you assess whether nanomaterials in cosmetics are safe? So FDA, is pursuing a pathway where they consider assessing the safety of cosmetic nanomaterials, and they intend to do that by considering various different characteristics and different kinds of studies. So these include physicochemical characteristics of potential nanomaterials, agglomeration and size distribution of nanomaterials under toxicity testing conditions and in the final product, impurities, potential routes of exposure to nanomaterials, potential for aggregation and agglomeration of nanoparticles in the final product, dosimetry for in vitro and in vivo toxicology studies, as well as in vivo and in vitro toxicological data on nanomaterial ingredients and their impurities, dermal penetration, potential inhalation, skin and eye irritation and sensitization studies, genotoxicity, and mutagenicity studies. So different testing methods and data need to be evaluated to properly address the function and unique properties of nanomaterials in cosmetics. And FDA is not so sure about traditional safety testing methods and whether they would apply to nanotechnology or nanomaterials in cosmetics. And so when considering different testing methods and the data that is needed to evaluate the safety assessment of cosmetic nanomaterials, you might really need to think outside the box. So FDA decides to start with really, detailing all different properties of nanomaterials. And so starting with the physical-chemical properties, they include the nanomaterial name, the chemistry abstract service number, structural formula, molecular and elemental composition, like including the degree of purity, known impurities or additives, particle size and distribution measurements, agglomeration and aggregation characteristics, density, solubility, surface chemistry, like surface coating, zeta potential or surface charge, functionalization, catalytic activity, changes in morphology, shape, surface area, surface topology, crystallinity, porosity, stability. So in addition to the physical-chemical characteristics, there is also the potential for nanomaterial impurities that could, be in the manufacturing process. So it could occur, nanomaterial impurities could be found, and it could be derived from the manufacturing process. They could be derived from manufacturing process changes, like the change to a solvent or temperature, time conditions, changes to materials, starting materials, or purity levels, or concentrations of the chemicals used in a manufacturing process, as well as changing the types and or quantities of other impurities in the final product. So you could easily manufacture a nanomaterial, and manufacturers need to be wary of this and determine also if other agents like dispersing agents and surface modifiers could also cause the manufacture of nanomaterials. So when a nanomaterial is manufactured from the manufacturing process or process changes, a manufacturer should strive to assess the identity and the quantity of those nanomaterial impurities and determine how they affect the overall safety of the cosmetic. So this can be done through toxicity characterization. So this, there could be routes of exposure that can be determined, dermal, inhalation, ocular, oral, toxicokinetic studies where we're looking at, absorption, distribution, metabolism, excretion, as well as toxicodynamic relationships where you're looking at dose-response relationships, as well as other other studies, whether it be acute toxicity, skin irritation, ocular irritation, dermal, photoirritation, skin sensitization, genotoxicity or mutagenicity, repeated dose, and subchronic toxicity studies. So finally, having discussed nanomaterials, and we're seeing that the manufacturing process is very important, both to ensure good, no potential contaminants, as well as no nanomaterial impurities that could affect the cosmetic safety. We really need some good manufacturing process practices for cosmetics. So cosmetic GMPs, or cosmetic good manufacturing practices, have they have been designed to ensure that the cosmetic products are manufactured consistently at a quality standard that is appropriate for their intended use. So it's concerned with both manufacturing and quality control procedures, and FDA's good manufacturing practices for cosmetics incorporates the ISO standard, ISO 22716, which was developed in Geneva, Switzerland. So the Federal Food, Drug, and Cosmetic Act, or the FD&C Act, prohibits the introduction or delivery for introduction into interstate commerce of cosmetics that are adulterated or misbranded. So that is Section 301. So therefore, cosmetic good manufacturing practices is pretty vital in ensuring that the Section 301 of the FD&C Act is complied to. So there are various facets to cosmetic GMP testing that together ensure the safety and quality of the cosmetics. So they include a lot of documentation all along the way in quality assurance. If something's not documented, it was not considered to be done. So there's a lot of record keeping in buildings and facilities, whether it be for manufacturing, packaging, storing. They're vital in GMP equipment, the equipment used for manufacture or for testing, the personnel, raw materials. As we know, they should not be contaminated, like water, color additives. Should they be batch certified or not? Do they have specifications, limits, cautionary statements? Are there any prohibited ingredients? There should not be. Are there any ingredients that are restricted? Do they comply with the various FDA concentration limits and other cautionary labeling? Also, production, what are the different controls for production yield, lab controls, internal audit for quality compliance? How are complaints handled? Is there a process for that? Are they all resolved? Were there any adverse events reported? Were there any recalls due to quality or safety issues? So we're going to be looking at some of the key areas in cosmetic GMPs as they relate to cosmetic testing. So equipment. So equipment that is used, especially measuring equipment, it should be calibrated. Equipment should be validated, and there should be a proper preventive maintenance program to ensure that the equipment is in a good working condition at all times. And this could incorporate manufacturer maintenance procedures and frequencies. So raw materials can be starting materials. They could also be raw materials used in packaging containers. So there could be sampling and testing to ensure the identity, purity, potency of all raw materials in accordance with pre-specified test methods, procedures, or protocols. And there can be test method validation to ensure that the test method is suitable for its intended purpose. Also, raw material suppliers can be audited to ensure that the raw materials that they supply are of the quality required for cosmetic manufacturing. And analytical specifications should be developed, and the tests should be, comply to the specifications. Water is a raw material, and it should be of a defined quality when it is used in cosmetics, and it should be tested and monitored for, according to chemical, physical, and microbiological specifications for quality. And that would be pre-specified. And there can be cleaning validation of equipment that supplies water. This is to ensure that there's no risk of water contamination or stagnation, which could lead to a biofilm buildup. And color additives, these can be, subject to FDA certification, as I discussed, or not. And then there is also prohibited and restricted ingredients, and we've covered these sections earlier, so I'm not going to get into that again. But these also would be subject to some kind of testing to determine concentration limits and so forth. Production controls. So selection, weighing, measuring raw materials, determination of the finished yield could be reviewed by a second individual, and there could be controls of each of these stages to ensure that the yield is as expected. And then major equipment, transfer lines, containers, and tanks that are used for processing, holding, or filling should be identified to indicate the content, the batch identification or designation, stage of the processing, and control status. This is important to ensure that there's no mix-ups that could otherwise be possible. And this would prevent potential contamination between different cosmetics or different ingredients that are manufactured in the same production facility or using the same production equipment. So there are also various measures that can be used to prevent contamination with microorganisms, extraneous material, chemicals, and so in process controls that ensure product uniformity, integrity, for instance, in-process batch weighing, or the accurate fill of mixing containers and adequacy of mixing. Various in-process controls should be considered at each stage of the manufacturing process to ensure that that batches are produced of having the right identity of cosmetic ingredients or cosmetics, and the right quantity, the right purity desired, so as to ensure that the cosmetic is not contaminated in any way. The theoretical yield for the production batch should be definitely compared to the actual yield, and this could be a good production control. Tamper-resistant packaging and labeling for liquid oral hygiene products as well as vaginal products should meet the requirements in FDA's Title 21 of the Code of Federal Regulations, Part 700, Section 25. And this is a regulation, and so it's not a recommendation. Also, packaging materials should be stored and handled in such a manner so as to prevent microbial or chemical contamination, and there should be no errors in product selection. Finished product packages should bear permanent, meaningful, unique lot or control numbers, and you can have a coding system that corresponds to these numbers. Lab controls, so for instance for raw materials like water, in-process and finished product samples, these can be tested in the lab for identity and compliance with specifications, and like physical and chemical properties. Microbial contamination tests can be performed according to the Bacteriological Analytical Manual of FDA to ensure that there's no microbial contamination, as well as there could be chemical contamination tests. All samples tested should be representative of a lot or a batch, and current finished product samples as well as retained product samples should be tested for adequacy of preservation against microbial contamination under reasonable conditions of storage and use. So approved lot samples of raw materials and finished products should be retained for an adequate period of time, and retained samples should be stored under conditions that protect their integrity, and there should be retesting at appropriate intervals to ensure continued compliance with established specifications. Return cosmetics, it should be examined, tested for deterioration, contamination, and compliance to specifications.